Meiosis is a type of cell division. The daughter cells have half as many chromosomes as the parent cells, making meiosis a reduction division.
This produces gametes, or sex cells, which only have half the number of chromosomes of a normal human cell, because two gametes, one from the mother and one from the father, will combine during fertilization to produce a zygote. Because gametes are haploid cells, the genetic variation increases with each generation, as the offspring is a random combination of both the mother and the father's genes.
First, interphase. The end of a cell's interphase stage prepares it for meiosis. In the S, or synthesis phase, chromosomes replicate through DNA replication, forming pairs of sister chromatids which are joined at the center. The physical point at which they are connected is called the centromere. This allows the first stage of meiosis to begin.
Prophase 1, DNA condenses to form chromosomes which become visible. Homologous chromosomes, meaning one from the maternal side and one from the paternal side, allow the genes from the mother and father to combine. Once the homologous chromosomes have paired up, they cross over, which ensures genetic variation for potential offspring.
For a slightly more detailed look at crossing over and its function, it is part of the reason why siblings, other than identical twins, never receive the same combination of their parents' alleles. The homologous chromosomes will line up, and parts will quite literally break off and attach to the other chromosome, transferring whatever genes were on those loci. As a result, a maternal chromosome will have some of the genes from the paternal chromosome and vice versa. During any crossover event, hundreds or even thousands of alleles are traded. They are transferred at the chiasmata, or the points on each of the chromosomes which stay in contact during the crossover. Crossing over affects an organism's genotype, thus encouraging the genetic variation key to evolution and adaptation. Back to the end of prophase.
Spindle fibers form from microtubules.
During metaphase 1, the bivalents, which is another name for homologous pairs, line up across the cell's equator. Then the nuclear membrane disintegrates.
It then moves into anaphase 1. Spindle fibers pull the bivalents towards opposite poles of the cell.
Then telophase 1 begins. In this stage, spindles and spindle fibers disintegrate and cytokinesis happens, where the cells split into two separate cells.
Were this mitosis, the process would be finished with two diploid daughter cells. However, as it is meiosis, it moves into meiosis 2, so that the further replication of these two daughter cells results in 4 haploid daughter cells.
The cell moves into meiosis 2. Conveniently, all the phases have the same names.
DNA condenses and spindle fibers form.
During metaphase 2, the nuclear membrane disintegrates, and the chromosomes line up along the equator in a process called random orientation. This means that the chromosomes do not line up in a specific order, thus allowing genetic variation. Then, the spindle fibers produced in prophase 2 attach to the centromeres of the sister chromatids.
In anaphase 2, the centromeres split to become individual chromosomes, and the spindle fibers pull chromatids towards opposite poles of the cell.
Random orientation means that the chromatids are not pulled in any particular direction.
The last stage is telophase 2. This is where chromosomes unwind and new membranes form for the cell to pull apart.
And there we have 4 haploid daughter cells. In males, these will go to form sperm cells, and in females, they will form egg cells.